Not all enzymes do the same thing. In fact, biochemists classify enzymes into 6 major classes based on the type of reaction they catalyze. Before we dive into examples, here are the 6 main types that you need to know. There is lyase, isomerase, ligase, hydrolase, oxidoreductase, and transferase. Now, obviously that's a lot to remember all at once. So, here's a mnemonic that can make that much easier. I present to you LIL hot, which is now your new favorite enzyme mnemonic. Each letter corresponds to one enzyme class. So, let's go through each one of them. First, we have lyase. Lyase breaks bonds without using water and without involving redox reactions. They often form double bonds or rings in the process. A classic example is aldolase in glycolysis. Just remember, no water, no redox, just bond breaking or forming double bonds. Up next, we have isomerase. Isomerases rearrange molecules into new isomers, meaning their molecular formula stays the same, but the structure changes. They don't add atoms, they don't remove atoms, they simply rearrange them. An example is malate isomerase. And as you can see, these structures are, if you had the formula in front of you, they would be the same, but they are obviously very different. Third, we have ligase. Ligases join 2 molecules together, typically using ATP. This drawing I have here doesn't really depict a specific reaction, just the fact that ATP conjoins the 2 circles together and leaves a byproduct of ADP. A good example is DNA ligase, which seals DNA fragments during replication and repair. Think ligate, like tying 2 things together. Fourth, we have hydrolase. Hydrolases break bonds using water. This is a hydrolysis reaction. For example, as depicted on the slide, lipase breaks down fats by hydrolyzing ester bonds with the help of water. Fifth, we have oxidoreductase. Oxidoreductases carry out oxidation reduction reactions by transferring electrons. A classic example, which is on the slide, is succinate dehydrogenase, which converts succinate into fumarate, which is actually a part of the Krebs cycle. During this reaction, 2 hydrogens are removed from succinate, forming a double bond. And at the same time, FAD is reduced to FADH2. And here's a helpful breakdown of the name. Dehydrogenase literally means to remove hydrogens. When you remove hydrogens, you're also removing electrons, because hydrogen carries an electron. So removing hydrogens is a form of oxidation. That's why dehydrogenases fall under oxidoreductases. Also, this enzyme is unique because it functions as complex 2 of the electron transport chain, linking the Krebs cycle directly to oxidative phosphorylation. Finally, we have transferase. Transferases move functional groups from one molecule to another. For example, kinases transfer phosphate groups. The reaction shown here is a general illustration of group transfer. It's not meant to represent one specific named reaction, just the overall concept that transferase moves functional groups from one molecule to another. On that note, let's review. Remember LIL hot. Lyase, isomerase, ligase, hydrolase, oxidoreductase, and transferase. If you understand what each class of enzyme does mechanistically, enzyme questions become much more straightforward, whether you're seeing them on MCAT or biochemistry or wherever you see them in your classes. Memorizing this makes your life so much easier. With that said, that's all I have for today. If you have any questions, comments, concerns I got something wrong, please leave a comment. And as always, like and subscribe, and I'll see you in the next one. Thanks.